Hello and welcome to Lyra Nara. In this video we're going to talk about a fascinating subject, and that is what really causes disease. And it's about misconceptions that are ingrained in our culture. It's about two competing theories, of which one became like a religious doctrine that is not questioned anymore, and the other one, despite great evidence for it being correct, got swept under the rug. Before we dive into that, if you enjoy this type of content where we talk about holistic health, please support our work by liking, sharing and subscribing, and also hit the notifications button, the little bell icon, so you don't miss out on the weekly content that appears here. Without further ado then, let's find out what this story is about and what really causes diseases. This fascinating story starts over 100 years ago at the end of the 19th century with two adversaries, Louis Pasteur and Antoine Béchamp. Louis Pasteur had the following theory. He said that the body is sterile and diseases arise from microorganisms that come from outside of the body, so from a pathogen invasion, from germs, bacteria and viruses that invade us. Microorganisms are generally to be guarded against, of course, because they are the culprits of diseases. The function of microorganisms is also constant, according to his theory. This is called monomorphism. The shapes and colors of microorganisms are also constant, and every disease is associated with a specific microorganism. And of course, according to Pasteur, microorganisms are the primary causal agents of diseases. And diseases therefore can also strike anybody. To prevent disease, we must build immunization with antagonistic vaccines. So the whole antibody theory, the immunization theory, that is still the popular theory nowadays in allopathic Western medicine. That all stems from the germ theory of Pasteur. And Western medical doctors even today work based on this belief, on this old-fashioned theory of germs and viruses that cause diseases. On the other hand, we have Béchamp. He experimentally studied pleomorphism. He identified the so-called microzymas that we discuss in more detail in the course of this presentation, and he's also the father of terrain therapy. So he believed the following. After the death of an organism, its cells disappear, leaving myriads of microzymas. And he not only believed it, but he was able to experimentally show that. Microzymas, according to Béchamp, can live on indefinitely after the decay of the animal's body or the plant they built on, and they are the only non-transitory elements of the organism. Microzymas are the building blocks of plants and animal cells which evolve into bacteria after the death of the plant or animal. Airborne organisms, known also as atmospheric germs or viruses, are actually microzymas, or the evolutionary forms of the bacteria set free by decompositions of the bodies in which they lived. Microzymas found by Béchamp in the natural chalk, but not those found in the artificial chalk, demonstrated for him that microzymas are the perpetuators of life and nothing is the prey of death. According to Béchamp, microzymas show functional differences for the same organ at different ages, in different organs of the same species, but also in different individuals of the same species. And this explains the variety of the diseases for different species, and also the fact that many diseases are not transmissible from one species to another, often not even from one individual to another within the same species. And this also explains why diseases are age-specific. By injecting exogenic microbes into plants, Béchamp was able to show that the bacteria identified under the microscope were other species than those initially inoculated. Disturbing the plant cells homeostasis induced microzymas mutation into this harmful bacteria form. So Pasteur thought and taught that germs cause diseases, that they never change. This is called monomorphism. While on the other hand, Béchamp thought and showed that diseases generate germs and that those germs can change. This is known as pleomorphism, and that the milieu, the environment in which they live in, causes these changes. We also have another third player here, Robert Koch. He postulated the most famous testing procedure that we're going to talk about shortly, and he stained bacteria and created also antibiotics. And he also believed, like Pasteur, that bacteria come from the outside and attack the body, and that they create toxins making you sick, and that of course they are also contagious. So let's see who was right according to scientific research that followed. In order to settle this discussion and see who is right, Robert Koch developed his 4 postulates to regulate the isolation and testing of germs, bacteria, and viruses that were not invented yet, so to speak, back then. They were not known. And these postulates, even today, are considered to be the golden standard for all microbiological tests. Interesting and important to notice is that none of the standards have ever been successfully applied to viruses, and this is the case even today. In 1930, electron microscope was discovered by Max Knoll and Ernst Ruska, and electron microscopes use electrons instead of light, and they can provide very accurate images of the smallest inanimate particles. The limitation is, however, that they cannot be used to study living things, because the electron irradiation destroys the viruses and the bacteria and the germs. Advanced light microscopes enabled also the observation of living organisms such as bacteria and their life in the human body. And it was with advanced light microscopes that it was clearly shown that bacteria do not create any toxins or poisons in a healthy living body. They only become harmful and create toxins when there is an oxygen deprivation, as it is the case, for example, in 2 to 4 day old cadavers. With advanced microscopes, scientists were also able to see that bacteria never produce toxins as long as the milieu, so the terrain in the body, is compatible with life conditions. The major toxins that poison the body are the own acidic metabolic waste. The main cause, one of the main causes, is stress, of course, also an unhealthy lifestyle, overeating, over drinking alcohol, other abusive addictions like smoking too much, over medication also with toxic pharma drugs, sedentarism, and one of the most important ones, the psycho-emotional burdens. Years later then, in the 1950s, 1960s, Canadian professor Gaston Naessens developed a tremendously advanced microscope which he used for researching diseases including cancer. And he studied a pleomorphism of live essential particles which he named somatids. Naessens' work brought more clarity in what Béchamp studied as microzymas. He found out that in healthy cells, the somatids that live in the tissue, the blood, and the intracellular cytosol of the cell are limited to 3 development stages only. They go through the somatid stage, the spore stage, and the double spore stage. First, there is a microcycle during which the reproductive hormone that permits cellular division is developed, and this healthy three-stage cycle is stopped by inhibitors in the blood during certain illnesses, including degenerative diseases. And this microcycle in 3 stages keeps going and becomes a 16-stage cycle during illness. When the cell environment is acidic, these tiny microorganisms pleomorphize into a 16 viral, bacterial, yeast-like fungus stages to help ferment the glucose excess and lactic acid that has resulted from the fermentation process. He said, little by little I was able to observe somatids and I extracted them from the blood. I was able to isolate it and grow it in a culture, and that's how I established its polymorphism. So a somatid is a basic living particle that is indispensable to life. Without it, cellular division cannot take place. But somatids are polymorphic. They can grow from one form to another, from a virus to a bacterium to a fungus. That means that if microbes, germs, or viruses are the result of diseases and not their causes, then treating, weakening, or attempting to kill those microbes or germs or viruses will not prevent or cure any disease. In 1918, Dr. Henry Lindlar mentioned in his book Philosophy of Natural Therapeutics that the physical characteristics and vital activities of cells and germs depend upon the soil in which the microzymas feed, grow, and multiply. Microzymas growing in the soil of procreative germplasm develop into normal, permanent, specialized cells of the living vegetables, animals, or human organisms. The same microzymas, however, that feed on morbid materials and systemic poisons develop pathogenic bacteria and parasites in the same living bodies. Dr. Lindlar continued to say that bacteria and parasites cannot cause and instigate inflammation or other diseases unless they find their own peculiar morbid soil in which they feed, grow, and multiply. End quote. Florence Nightingale, the great pioneer of nursing, noted also, along with Dr. Creighton and Farr and many other doctors, that infectious diseases replace each other according to varying degrees of unhealthiness of the living conditions of the people. For example, the plague was replaced by typhus and smallpox, and later on by measles. So there are no specific diseases, there are specific living conditions that cater for these diseases to develop. For example, today we have cancer epidemic, we have Lyme disease and its many co-infections, we have a diabetes epidemic, and those are decimating millions of people, and this comes from the damaged milieu that we have created within our bodies, from all the toxins, from the wrong nutrition, for the sedentarism, etc., according to the terrain therapy theory. So is there also more recent science that supports this milieu theory of Béchamp? Well, we have, for example, the work of Dr. E.C. Rosenau. He is the earliest record we have of the corroboration of Béchamp's theory. In 1920, at the Meyer Biological Laboratories, Dr. Rosenau took bacterial strains from many different disease sources, such as perpetual sepsis, arthritis, tonsillitis, and cow's milk, and put them into one culture of a uniform media. And after a while, there were no differences between the germs. Thus, Rosenau concluded that there was no particularly fixed species of different germs, and all of them have the capacity to change their structure with the changes of nutrients. It would seem, therefore, that focal infections are no longer to be looked upon merely as a place of entrance of bacteria, but as a place where conditions were favorable for the bacteria or the germs to acquire the properties which give them a wide range of affinities for various structures. And this study was published in 1914 in the Journal of Infectious Diseases, by the way. But those are not the only scientists that prove Béchamp's theory right. No, the truth of Rosenau's ideas and Béchamp's ideas was proven also in countless other instances in sanitary practices. For example, William Miller tells us the following. When typhoid fever was discovered to come from contaminated water, pure water quickly eliminated typhoid fever. The same is true for puerperal fever, which killed so many women during childbirth. Semmelweis had also a hard time to convince physicians that they spread the diseases with their contaminated hands and instruments during surgery. And many infections, of course, were eliminated as soon as such sources were removed by disinfecting the instruments before surgery. Asher reported also transforming cocci, the round-shaped bacteria, into bacilli, the long-shaped species. The initial infections with pus show also streptococci, round-shaped bacteria, and in the later stages these traps are converting themselves into staphylococci due to the progressing disintegration of the blood cells. Again, a proof for the pleomorphism theory. Victor Henry also transformed anthrax bacteria into a spherical coccus by using UV light. Dr. M. V. Gallipi figured out that the crushed tissues of a weapon wound favors the appearance and evolution of certain intracellular bacteria. The extravasated blood may give birth directly without foreign collaboration to infectious elements, so an absolutely sterile projectile even can trigger infections by its mechanical action, which changes, of course, the milieu around the wound. Disease is born of us and in us, and it depends on the milieu. Béchamp also concluded that microzymas are indestructible. They are endowed with perennial life. Dr. Royal Rife also played an important role in proving Béchamp right because he built a very advanced light microscope that back then was even more capable than the first electron microscopes that also appeared around that period. It had a magnification power of 150,000, and with this advanced microscope Rife was able to make germs visible, and also demonstrated that by altering the environment and the food supply, friendly germs such as column bacillus can be converted into pathogenic germs such as typhoid, which is also considered to be a cancer precursor. And he also showed, interestingly enough, that this process is reversible when you change the milieu. Rife stated, quote, "In reality, it is not the bacteria that produce the disease, but we believe it is the chemical constituents of these microorganisms enacting upon the unbalanced metabolism of the human body that is actually producing the disease. We also believe if the human body metabolism is perfectly balanced or poised, it is susceptible to no disease." Again, germs are part of the disease process rather than the instigator of this process. He could also, with his microscope, look at the smaller cells, the microzymas, in the smallest body cells. So he really experimented with this and was able to see it happening live under the light microscope. And this might also explain why all of the people who are dying now from COVID-19, almost all of them have serious preconditions. So their milieu is deranged, it's distorted, it's unbalanced already. And this makes it possible for the virus to become harmful. There were even experiments during the Spanish flu pandemic that showed it to be non-contagious, believe it or not. So let's look at this. Navy doctors, for example, collected the mucus from men who were desperately ill from the Spanish flu, They gathered a thick viscous secretion from their noses and throats, and they sprayed this mucus into the noses and throats of some men and dropped it into other men's eyes. Of course, those men were healthy. And in one attempt, they swept mucus from the back of the nose of a man with the flu and then directly swabbed one patient's nasal septum and rubbed it into the nasal septum of one of the volunteers. And they tried to simulate what happens naturally when people are exposed to flu victims. The doctors took then 10 of the volunteers into the hospital ward where men were dying because of the Spanish flu. And the sick men lay huddled on the narrow beds, burning of course with fever and drifting in and out of sleep in a delirium. So they were really seriously sick. The 10 healthy men were giving the instructions. Each was to walk up to the bed of a sick man and draw near him, lean into his face, breathe his fetid breath, and chat with him for 5 minutes. And just to be sure that the healthy man had a full exposure to the sick man's disease, the sick man was to exhale also deeply while the healthy man drew in the sick man's breath directly into his own lungs. Finally, just to make it doubly sure, the flu victim also coughed 5 times in the healthy volunteer's face. So what do you think happened? Each healthy volunteer repeated these actions, by the way, with 10 different flu patients, and each flu patient had been seriously ill for no more than 3 days, which is the period when the virus or whatever it was that was causing the flu should still be around in his mucus, in his nose and in his lungs. But not one single healthy man got sick. There was also a comparable experiment carried out under much stricter conditions. It took place in San Francisco with 50 imprisoned sailors, but once again the results did not correspond with what the doctors had expected. Were those the only studies on contagiousness of viruses? Not by far. There were also experiments, for example, by the New York State Health Department and the United States Public Health Service on polio, and it showed clearly that polio was not contagious. So what did they do? During one of the most widespread polio epidemics in 1949, they studied the contraction of polio by definite contact with other victims of the disease, and they could not establish it. The United States Public Health Service in its studies also found the same negative answer during succeeding outbreaks of polio. So they did this more than once. In short, they learned that the disease was not contagious. Time magazine even commented on this unexpected revelation of these surveys by remarking that, quote, "when and where people catch polio remains a mystery," end quote. So the supposition that viruses entered through the mouth and nose led the experimenters to make exhaustive tests on both animals and human beings, mostly prisoners and orphans in institutions. And those people were exposed by close association to polio patients, and their throats and nasal passages were frequently swabbed with matter from the sick patients that were supposed to contain the viruses, of course. And no polio was produced in this way, and about the only noticeable after effect was that the subjects of the experiments lost their sense of taste and smell due to the damaging effects of the poisons in the serum that was used on the swabs. This is, by the way, from a very interesting book that is called The Poisoned Needle by Eleanor McBean. This is in chapter 10. And it's on the hidden dangers in the polio vaccine. Another extract from another interesting book that is called Exposing the Myth of the Germ Theory. This is written by Arthur M. Baker. And he states, quote, "Take colds for instance. How is it that infants have about 8 colds per year while the parents only a few? How is it that those persons isolated at observation posts in the North and South Poles catch colds during their stay? How is it that between 1965 and 67 the National Institute of Health cold laboratories in Bethesda, Maryland conducted experiments that showed everything but contagion? Volunteers were swabbed daily with supposed cold viruses taken directly from those suffering colds, and none of them became ill. In the meantime, shortly following traditional Thanksgiving feasts, the number of colds in both groups increased dramatically, as would be expected when you consume excessively rich food and a lot of drinks, as it happens during holiday festivities. Venereal disease is also supposed to be contagious, but the so-called contagious factors, or the bacteria, are present because of the disease and are not the cause of the condition. And 20% or so of those suffering venereal disease have neither gonococcus nor spirochetes, when those are the ones who are said to cause the disease. Also think about the following. We constantly breathe in about 14,000 germs and bacteria per hour per day. If germs are so harmful, why aren't we all dead? With all that in mind, let's try to redefine what a virus is then. So viruses are non-living protein structures, in contrast to bacteria, parasites and fungi, as well as human tissue. Viruses do not have their own metabolism. They do not have their own proliferation mechanism. They do not have their own digestion. They share RNA and DNA with specific cells. And therefore viruses originate in the presence of a living cell to help eradicate the toxins. Because of these characteristics of viruses, it cannot be viral contamination. It cannot exist. Viruses are not contagious and cannot spread, as we have seen also from the experiments that were performed, from one organ to another organ, and neither from one individual to another. Even less so from animals to humans. Viruses rely on very specific RNA and DNA that is shared by the bacterial cell, which is called for help, so to speak. Viruses also do not invade every kind of cell in a body, but only very specific toxic cells from which they got the RNA DNA as an energy vector to eliminate the cellular debris, the so-called zeta energy potential. Viruses orient themselves via antibodies. For example, hepatitis viruses cleanse the liver, and based on the degree of toxicity, we identified different types, such as the A, B, and C hepatitis viruses. Coronaviruses cleanse the lungs and the lower respiratory ways. Rhinoviruses and adenoviruses cleanse the upper respiratory airways. This is where you get the cold and the flu from. Viruses eliminate the toxins via skin, mouth and nose, also the urinary tract and the gastrointestinal tract, so via urine and via feces, and therefore they are found close to these organs and in these excrements. After viruses biologically shredded, so dissolve the toxins, white blood cells expel then the cellular debris. And viral infections reverse cellular toxicity to save cells and keep living bodies alive actually. Intense cellular toxicity means high viral activity. When toxins are being broken down, there are symptoms which intensify with the virus concentration. And people may even die during this process if there is an excessive toxicity, thus intense viral activity, and if they are also additionally medicated with very aggressive toxic drugs such as antibiotics, painkillers, vaccines, etc. It is the same, by the way, in cancer when tumors, so bags with toxins, are surgically removed, but thereafter the body is being toxified to death with chemo drugs or radiation. Nature and living beings and humans and viruses act in cycles. They are influenced by weather, temperature, humidity, daily light changes, and nutrition. And therefore the overload of toxins in the blood is also cyclical. And this explains why you see the flu season, for example, because the flu viruses are always busy in the fall and spring. And in these pictures you can see how a virus is created. The proteins that already exist in the host cell enter the nucleus, where the cell then manufactures the virus. The virus is manufactured in the host cell, is then released from the nucleus and is ready to exit the cell. And the virus is replicated in the same manner as the first step. So here in the pictures on the right, you can see the bursting of a cell to release these viruses of type 1 to dissolve the cellular debris. And in the second picture, you see the bursting of a cell to release the viruses type 2 to cleanse the cellular debris that is left over from the first virus. So this whole virology theory, this whole germ theory, as you can see, has a mountain of evidence against it. It's very problematic, and unfortunately we base a lot of wrong treatments and conclusions on that, because it has become doctrine, a religious doctrine that cannot be questioned despite this evidence against it. Also, tests like PCR cannot determine the amount of viral replication that occurs in the body. We've talked specifically on the problems with the PCR test when testing for COVID-19 in a separate presentation. Please check it out. We go there into much more detail why this test is very imprecise and gives you a lot of false results. And also, the virus, the SARS-CoV-2 that is supposed to cause COVID-19, has never been isolated. Postulates have never been fulfilled for any virus for that matter. PCR test may observe the person that the person has viral genetic material, yes, so some fragments, some RNA fragments, but this does not mean that there is an active virus that makes this person sick and causes the symptoms. These COVID-19 masters today can mask the cases by claiming it is all COVID-19, and it could be any coronavirus strain for that matter, which could be claimed to cause COVID-19, because there is literally no difference between COVID-19 and other corona strains when looked through a PCR test. And actually the test will not see the difference because there is not a big enough difference to note. There is always some level of viral and bacterial activity in small amounts that works in the body. That is true. It is only when large amounts of viral and bacterial activity are called upon that the body experiences symptoms, however. All bacteria and all viruses, all germs are good if they occur in the body naturally and in a healthy milieu. Both are used by the body to heal and restore itself. So it's a symbiosis, it's not a fight. It is not that it isn't effective enough, it's that the body cannot rationally deal with large amounts of toxins in the blood, and viruses break down those toxic substances. If the body does not have the right amounts of fats and alkalizing minerals such as potassium and calcium to bind with the toxins, or if the liver or the kidneys are compromised, toxicity can lead to death because the immune system cannot deal with this overload. Also keep in mind, as we've seen, a virus is not alive. It has no respiration or nucleus and no digestion, and it also cannot replicate on its own because it's not alive. That's like saying a rock can replicate itself into two rocks, etc. And therefore viruses can never occur in nature magically by themselves. They must have a living cell to call for help, with which they share then the RNA DNA that is specific for that cell, and only this way they can start to replicate. And this one point alone invalidates the entire idea behind viruses being contagious and also creating epidemics and pandemics. If viruses must have a host cell to replicate, they cannot do so outside the body on their own. And therefore, for them to originally appear in nature, they would have had to have come from a cell. If they come from a cell, it invalidates the theory that they infect the body. Is our way of thinking today that infectious diseases are the result of a choice that was intentionally made around the middle of the last century? Was that choice the result of a fork in the road in which it took the wrong path? Was this road taken because of financial interest and aggressive Rockefeller and Carnegie Foundation capital injections? And because it was more beneficial to commercial exploitation? Well, answer these questions for yourselves. You can also involve yourselves into the comment section below the video and discuss it amongst yourselves. But the truth is that one theory became religious doctrine, and the other one, the milieu theory, for which there is so much evidence and with which you could explain all these strange things that are observed now also in the COVID-19 pandemic, you could explain it, in our opinion, much better. So that's it for this presentation. I hope that you've enjoyed it. If you did so, please support our work by liking, sharing and subscribing, and also hit the notifications button. And until next time, stay healthy.